on the agenda. And what we're going to be doing is next is announcing the poster winners. And so I think what I'm going to do is do this slightly differently than we've done in the past. And what I'm going to do is I'm not going to tell you who has won first, second, and third place. But what I'm going to do is to invite the first, second, and third place winners up to the front. And we'll have you all up here. So the first person I'd like to invite up is Katayun Kasayan. Did I pronounce it? Am I close? Well, she's coming to the front. Gia Wing. Gia Wing. And lastly, Mark Hills. So I think what we're going to do, maybe we'll just do it in the order that you are right here, is we're going to give you, let's say, 30 to 60 seconds each, just to give a quick run through on your project. Just tell us, and you can go up to the microphone here, and just tell us about what you've done. Oh, sorry, you can use that. You can do it karaoke style. And so please just spend 60 seconds and just tell us about your project, and then I'm going to announce first, second, third place winners.
So my poster was the outline of the project, personalized genomics that's been going on at the BC Cancer Agency for the past year and a half. A big group of scientists from the Genome Sciences Center, many of in the room today, as well as a big group of oncologists are involved in this project, not just the representative. Late-stage cancer patients that have gone through every possible therapy that they could have gone through, and they have no options yet, and they're progressing and come through the program. We sequence the genome and the transcriptome of the tumor samples as well as blood, and try to figure out mutations and targets that could possibly be targeted to be available, sometimes approved, sometimes in clinical trials.
Thank you. Katayun's project is entitled Bioinformatic Analysis Approaches for Personal Oncogenomics. Thank you very much. Gia.
So I studied B cells of the immune system, and B cells are antibody-producing cells that protect you from infections. And I studied how the cytoskeleton can work together in promoting B cell activation, and I use a number of different RNAi approaches to knock down regulators of the cytoskeleton, specifically the actin cytoskeleton as well as the microtubule cytoskeleton. So what I've showed in my poster is that there's a GTPase protein that acts as a master regulator of cytoskeletal dynamics, which is called RAP, and RAP is involved in polarization of the cytoskeletal system during B cell activation when it recognizes viral or bacterial pathogens. So, thank you.
And I'll just mention that the title of her poster was B cell receptor induced reorientation of the microtubule organizing center to the B cell immune synapse requires RAP activation and actin reorganization. Brilliant title. So our last is Mark Hills, and the poster was entitled Leapfrogging Genome Assemblies by Sequencing Single Cells. So, Mark.
Yeah, so I'm kind of using a very random single cell approach to better assemble genomes. Essentially, it gives us a way of labeling a chromosome with a specific signature. So if you take a scaffold genome that consists of maybe 200,000 fragments, we can actually pass those fragments back into which chromosome they belong, and also order them into a better built genome.
Excellent. Okay, so the moment you've all been waiting for now is our first place winner is Mark. Mark. You're a second place winner. Thank you very much. So let me just say that we've had 35 posters were presented. We have a wonderful group of people that volunteered to be judges today. And I mean, all 35 of you deserve great congratulations. You've all done just a superb job. But special congratulations to our winners today. So, and let me just mention to you guys now, we're going to make you do a little bit more work. So, so the first place winner, Mark, $500, $300 to Gia, and $200 to Kategan. And what you're going to be doing, though, is you're going to be coming by Genome BC, and we're going to organize this with you. And you're going to talk to our staff, and you're going to give our staffs just about a 5-minute run by, and then we're going to present the check to you. So a little but we'll even buy you a coffee. So, but please join me again in just. So folks, today, you know, I hope that you've all enjoyed the program today. I mean, the program today is your program. You've all asked us to change things a bit, and we've taken these comments to heart. I think this has been wonderful. I thoroughly enjoyed it. Everything that's been going on and everything's been positive. So again, just thanks to each and every one of you for making the day the success it has been. I need to give some special thanks, though, to a number of people. So I'm just going to mention a bunch of names here. So Sally, Cease, Catherine, Julia, Damien, Sheila Driscoll, Catherine Jarvis, Mary McKay, Gabe, Rachel, our chairman, Alan Pellman, the Student Biotech Network. I mean, you guys have been just great. Are all the judges of the poster competition. Thank you all very, very much. Probably many of you didn't even get a chance to have a bite to eat, so thank you for that. And thank you to all of you for just attending today and for your comments. And we're going to look for your comments for next year's program, and we're going to pay close attention to those, and we'll try to make this work the best we can for you. And last but not least, let me just mention, I think that if I didn't get my cliff notes, so I hope I'm right. I'm looking for the communications group here. But I think the next event is our summer barbecue, which is July 16th. And that'll be being announced very soon, the venue for that. But it'll be the same venue as we've always used. It'll be 11:30 to 1:30, and so we hope to see you all there. So again, thank you very much to all of you and to our speakers, to attendees, to our winners today, to all the other poster competitors, and thanks to all of you. We'll see you again soon.